Good evening, viewer. Welcome to our Glory Globe TV's breaking news. Yeah, researchers have studied the process by which phages, viruses that infect and replicate within bacteria enter cells for over 50 years. In a new study, scientists from the University of Illinois Urbana-Champaign and Texas A&M University have employed cutting-edge techniques to examine this process at the single-cell level. The field of phagobiology has seen an explosion over the last decade because more researchers are realizing the significance of phages in ecology, evolution and biotechnology, said Ideo Golding, a professor of physics. Yeah, this work is unique because we looked at phage infection at the level of individual bacterial cells. The process of phage infection involves the attachment of the virus to the surface of a bacterium. Following this, the virus injects its genetic material into the cell. After entering, a phage can either force the cell to produce more phages and eventually explode, a process called cell lysis, or the phage can integrate its genome into the bacterial one and remain dormant, a process called lysogeny. Yeah, the outcome depends on how many phages are simultaneously infecting the cell. A single phage causes lysis, while infection by multiple phages result in lysogeny. In the current study, the researchers wanted to ask whether the number of infecting phages that bind to the bacterial surface correspond to the amount of viral genetic material that is injected into the cell. To do so, they fluorescently labeled both the protein cell of the phages and the genetic material inside. Then grew Escherichia coli, used different concentrations of infecting phages and tracked how many of them were able to inject their genetic material into E. coli. Known since the 70s that when multiple phages infect the same cell, it impacts the outcome of the infection. In this paper, we were able to take precise measurements unlike any study done so far, Golding said. Researchers were surprised to find that the entry of a phage's genetic material could be impeded by the other conflicting phages. They found that when there were more phages attached to the surface of the cell, relatively fewer of them were able to enter. But data shows that the first stage of infection, phage entry, is an important step that was previously underappreciated, Golding said. We found that co-infecting phages were impeding each other's entry by perturbing the electrophysiology of the cell. The outermost layer of bacteria is constantly dealing with the movement of electrons and ions that are crucial for energy generation and transmitting signals in and out of the cell. Over the past decade, researchers have started realizing the importance of this electrophysiology in other bacterial phenomena, including antibiotic resistance. This paper opens a new avenue for research in bacterial electrophysiology, its role in phagobiology. BNS exclusive report, Glory Globe TV, Delhi, Bharat.